Our next topic is morphology and physiology of bacteriology. Morphology and physiology of bacteria. So let us first dive into morphology of bacteria. When we talk about morphology, firstly we have to know there is a gram staining, which is the important staining method. According to gram staining, we classify the bacteria as gram positive and gram negative bacteria. Gram positive bacteria are those bacteria which take the primary stain, that is crystal violet, and gram negative bacteria are those bacteria which will not take the primary stain, that is crystal violet, but later on they take the secondary stain, that is sofranin. So gram positive bacteria are violet in color, whereas gram negative bacteria are pink in color. So gram positive bacteria and gram negative bacteria can be either be cocci or bacillus. Okay. So let us dive into the different shape of gram positive and gram negative bacteria. So you see, this is the gram. First we classify gram positive bacteria based on different shapes. So gram positive bacteria can be seen as clusters. For example, Staphylococcus. And they can be seen as chain. For example, strepto means chain, streptococcus. Staphylo means group. Okay, streptococcus, staphylococcus. Then they can be seen as tetrad for individual coccus as a group, that is, for example, micrococcus. Then they can be seen as octate. Octate is usually not seen like this. Octate, there will be a tetrad and behind them another 4 will be there, totally 8. For example, our Sarcina. Then they can be seen as pairs. For example, our pneumococcus and enterococcus. Okay, these are the gram positive cocci. Then let us dive into gram negative cocci. Gram negative cocci, our famous Neisseria species, Neisseria, gonorrhea and Neisseria meningitis, or we also call as gonococcus and meningococcus. Then let us dive into bacilli. Bacilli means they are rod shaped structures. There are gram positive bacillus and gram negative bacillus. So gram positive bacillus as in chains, example Bacillus anthracis. Bacillus anthracis. Okay, Bacillus anthracis. Bacillus in chain, for example, Bacillus anthracis. Then they can be cuneiform pattern. Cuneiform pattern means V letter, English V letter size ആയിരിക്കും, or they are also similar to Chinese letters. So cuneiform, example, our famous diphtheria. Corynebacterium diphtheria is the famous example for cuneiform pattern gram positive bacillus. Then there is gram negative bacillus. Gram negative bacillus example Proteus. I will show you the real slide of these. Then there is comma shaped gram negative bacillus, comma shaped Vibrio. We also know Vibrio means comma. So our Vibrio cholerae is a comma shaped gram negative bacillus. Then there is the bacteria which will not take the gram stain, for example spirochetes. They have the spiral shape. You see, these are the spirochetes. Here special stain is used. The special stain used here is silver impregnation method. We will detail this study about it in later. So these are the spirochetes. See, spring like structures. So these are the different shapes of bacteria. Let me show you the slide. Okay, you see, this is the micrococcus seen as tetrads. And these are the staphylococcus. You see, staphylococcus means there will be clusters. You see, gram positive cocci arranged in clusters. These are one cluster, these are the other cluster. You see, these are gram positive cocci, for example staphylococcus. Then this is our tetrads. You see, they are arranged in 4. See, there are 4. These are tetrads. For example, micrococcus, we already taught. Tetrad example, micrococcus. Then there is octate arrangement, that is one tetrad and behind them is the other. These are example for octate shape. These are kind of octate. Our Sarcina is the example for octate. Okay, these are gram positive cocci are this. Then there is gram positive cocci arranged in chains, that is streptococcus. Staphylococcus are in clusters, streptococcus are in chains. Okay, then there is gram positive bacillus, our bacillus anthracis, which has an indigenous spores. Okay, gram positive bacillus, rod shaped arrangement as in chains, bacillus anthracis. Then there is gram positive bacillus which are arranged as a branching filament. These are actinomycetes. You see there is a branching from bacillus. You see branching is there. These are actinomycetes, gram positive branching filaments. Then is our famous Corynebacterium diphtheria, which are cuneiform in shape. You see there are Chinese lectures, V-shape arrangement, arranged as V-shaped or also known as Chinese letters. These are cuneiform Corynebacterium diphtheria. You see these are Corynebacterium diphtheria. This is the systemic cell, and in systemic cells, blood cells, we can see V-shaped arrangement, cuneiform cells. Okay, see V-shaped arrangement, cuneiform cells. These are Corynebacterium diphtheria in blood smear. Then there is gram negative bacilli. Example our proteus. These are gram negative, kind of pink in color, pink or reddish in color. Example proteus. Then these are gram negative cocci. Our meningococci and gonococci are example for gram negative cocci. You see, we can see gram negative cocci in white blood cells. These are WBCs. These are blood smear. In blood smear there is gram negative cocci. These are Neisseria gonorrhea. Okay, so these are all the pictures of real slide. The next morphology, that is cell wall. So in cell wall, also known as O antigen. Okay, so function is protection. We already know. And the cell wall, we have to talk about gram positive, gram negative bacteria, mycoplasma and mycobacteria, and also about fungus. So firstly, when we talk about gram positive, we already know gram positive bacteria take out the primary chain itself and will not remove after we adding a decolorizer when we are doing gram staining. So that means it has a thick cell wall. Okay, so gram positive bacteria has a thick cell wall. So the gram positive bacteria cell wall are made up of peptidoglycan plus teichoic acid. Okay, this is thick layer. Peptidoglycan is a thick layer. About 100 layers. There are about 100 layers. And when we talk about this is one of the reason why it takes a primary chain, that is crystal violet. When we talk about gram negative, they also have peptidoglycan, but it is thin. Peptidoglycan layer is thin. And they have, instead of teichoic acid, they have lipopolysaccharide. This is about 10 layers only. And there has about lipopolysaccharides, which is one of the reason why they do not take primary chain, that is crystal violet. But later on, after adding decolorizer and later when we are adding sofranin, they take out the sofranin dye. That is why they are pink or red in color. So gram negative have peptidoglycan and lipopolysaccharide. Then we have to talk about mycobacteria. Mycobacteria is an example for what? Positive acid fast. Okay, because it take out the acid fast chain. And the reason why it is an acid fast is because due to its cell wall content. The cell wall of Mycobacterium has peptidoglycan. Then there is mycolic acid. Okay, this mycolic acid is the reason why it is acid fast. And then there is also lipoarabinomannan. Lipoarabinomannan, lam. Okay, so this is the content of mycobacteria, and it is acid fast due to mycolic acid. Then last we have to talk about fungal cell wall. We already know fungal cell wall is made up of chitin. Okay, then how to demonstrate? These are the cell wall content of gram positive, gram negative mycobacteria and fungal cell wall. Then how to demonstrate? How to demonstrate cell wall is by mainly three methods. The first method is electron microscopy. We can see cell wall in electron microscopy. Then there is micro dissection, and then third method is plasmolysis. Plasmolysis is nothing but when we keep bacteria in hypertonic saline. You see, we already know what you mean by that, no? When we keep in hypertonic saline. So what happens is that if this is the cell wall and this is the cell inside, when we keep in hypertonic saline, what happens is that the cell membrane will shrink here, and cell wall can be easily identified. Here plasmolysis okay. Okay, hypertonic saline. So these are the three methods of demonstrate of cell wall: electron microscope, micro dissection, plasmolysis. And we have to know why gram positive bacteria take the primary strain, because that has a thick peptidoglycan layer, and in gram negative there is lipopolysaccharide and a thin peptidoglycan layer. That's why it will not take crystal violet. And then there is mycobacteria, which is an acid fast due to mycolic acid. Then there is fungal cell wall, which is made up of chitin. Next we talk about cell wall appendages. Okay, third one is cell wall appendages. So cell wall appendages are nothing but which present outside the cell wall, like capsules, slime layers, flagella, pili, fimbriae, etc. Okay, so these are the cell wall appendages. So first we deal about capsule and slime layer. These are nothing other than glycocalyx or glycocalyx. Okay, they are glycocalyx. Glycocalyx are what? Amorphous. They are amorphous viscid material outside cell wall. So capsule and slime layer are glycocalyx. So glycocalyx can be either be capsule or slime layer. If it was capsule, then it is tightly packed. And if it was slime layer, what slime layer will be loosely packed. It can be easily washed away. So these are slime layers. Loosely packed glycocalyx are called slime layer. We have importance for capsule. So capsules are also known as what? They are also known as K antigens. Capsule antigens are known as K antigens. Now let us discover the function. What are the function of capsule? Slime layer has not that much importance, but capsule has very much importance because it is one of the important virulent factor in many bacteria. Okay, so first function is there are antiphagocytic. You see, capsules are antiphagocytic. That means what? Phagocytosis is the first line of defense. First line of defense is phagocytosis. So antiphagocytic means it escapes the first line of defense of our immune system. So that's why it is one of the important virulent factor. So these are the functions of K antigens. Now let us get into the examples of some of the capsulated bacteria. Okay, so it includes our famous meningococcus, our pneumococcus, then our bacillus anthracis, then our Haemophilus influenzae, then our Klebsiella, and the sixth one is our Clostridium perfringens. Perfringens. Okay, perfringens. These are the our six bacteria which has capsules. So these are our important virulent factors. Now let us study about how we demonstrate the capsule. So basically there are two methods to demonstrate the capsule. The first one is our staining normal methods. There are two types of staining method. And the another method to demonstrate capsule is immunological reaction. Okay, immunological method. So first we deal about demonstration. How to demonstrate capsule? There are two methods. Okay, the first one is our staining method. Okay, in staining method there are two types of method. One is our negative staining. In negative staining what happens is that it stains the background and inside the bacteria, but it will not stain capsule. So there will be a halo appearance. That means a balance. That means a blank appearance around the bacteria. That is called a halo appearance. So the important negative stain techniques are our important stains are India, Inga, and the necrosis. Necrosin is the best, but it is so costly, so we mostly commonly use our India ink. Okay, so what happens here is that in negative staining, if this is a slide and this is a bacteria, then this is the capsule. You see, capsule will not get stained by our negative stain, but the background and inside the bacteria gets stained. So there will be an appearance of what? There will be a halo appearance. This is called a halo appearance. So in negative staining, there is a halo appearance which is capsule. Capsule will not get stained. Then the next staining technique in to demonstrate our capsule is nothing other than McFadden's reaction. This is reaction. Okay. So McFadden reaction is specific to Bacillus anthracis. McFadden reaction. So McFadden reaction is specific to capsule of Bacillus anthracis. The reason why is that is because Bacillus anthracis, shit, sorry, Bacillus anthracis has polypeptide capsule. So McFadden reaction is only possible in Bacillus anthracis specific to Bacillus anthracis. The reason why which is that the stain here used is our normal crystal violet. Crystal violet stain is used and it only stain this crystal violet only stains polypeptide capsules. Okay, so McFadden's reaction is specific to demonstration on capsule in Bacillus anthracis, but negative staining usually do in all capsules. Okay, then the next demonstration technique other than staining method is our immunological method. There are two types of immunological methods. Immunological methods is nothing but you will see immunology is related to antigen and antibody, right? So we already told capsule is called as antigens. They are act as antigen, that is the virulent factor. So we demonstrate those antigens by immunological methods. So there are two types of immunological reactions that we use. First one is the lattice agglutination. Lattice agglutination method. Here what we do is that on a latex we coat with anti-capsular antibodies. Okay, So when we add the serum that contains this capsule containing bacteria, there will be an agglutination reaction. That is called lattice agglutination method. Nothing but here what happen is that our antibody coated on latex plus our capsular antigen add ചെയ്യുമ്പോൾ there will be agglutination. That is latex agglutination method. And the next immunological method is our quilling. Quilling's reaction. Quelllengs. Okay, Quilling's reaction. So what we do here is that there will be antibody against capsule, capsular antigen. Okay, against capsular antigen. So when we add antibody against capsular antigen into the serum containing capsule, then the capsule swells. Capsular swelling will occur. This is called Quilling's reaction. Here antibody is not coated in the lactase. Here just the anti-capsular antibodies are added, so the capsule gets swollen. That is called Quilling's reaction. Okay. Okay. Now let us discuss about the second cell wall appendages, that is flagella. Okay, flagella is also known as H antigen. So the protein the flagella is made up of is flagellin. Protein is flagellin. Function, we already know function is locomotion. Then talk about structure. So when we talk about structure, flagella consists of three components. That one is the basal body, which is the complex part of flagella. It is embedded in cell membrane and it is made up of 2 to 4 rings. Okay. Then there is hook, which is flexible, and then there is filament that helps in locomotion. This is the structure of flagella. Then how to demonstrate? The next one we have to talk about is demonstration of flagella. So there are two methods in demonstration. One is the direct method and another one is the indirect method. In direct method, obviously we have electron microscope, because electron microscope will show the exact shape and image of flagella, electron microscopy. Then the second one is flagella staining technique. Okay. So the flagella staining method we use here is Leifson's staining method. Okay, Leifson's staining method. It is a type of tannic acid impregnation method. Tannic acid staining method. We will detail this study about impregnation staining and special staining in another class. Okay. So we uses one is the Leifson's staining method. Then there is indirect method. So the first one in the indirect method is our special microscopy. We already told that in dark field microscopy and in phase contrast microscopy we don't require staining method. Okay, so staining, non requirement of staining means there will be live individual. We are demonstrating live individual, so we can appreciate the motility of the organism in dark field microscopy and phase contrast microscopy. Then the second technique we use here is hanging drop method. We already learned about hanging drop, how to demonstrate it. It is found by Louis Pasteur. Okay, hanging drop method, because here we can demonstrate the motility of an organism under normal microscopy. Okay, then there is another method, wet mount preparation. Then there is semi-solid agar. Here we use one is the mannitol agar. Okay, So you see, unlike our solid agar culture, in semi-solid agar, the bacteria can grow in depth also. Okay, so if it was a motile organism, then it can move downwards too. When forming colony, there will be having a depth in semi-solid agar, so we can demonstrate motility in semi-solid agar. Okay, then there is our fifth method, because we obviously told flagella is an H antigen, so there we can demonstrate anti-flagellar antibody. H antigen can be demonstrated by anti-flagellar antibodies. So these are the indirect methods how we can demonstrate flagella. Then our third, cell wall appendages are fimbriae, or also known as pili. Okay, so fimbriae or pili. Fimbriae is called as F antigen. Here the protein is protein that made up by pilin. Okay, not a fimbrine, it is pilin. The protein is pilin. Function of the fimbriae is adhesion. Small hair-like structures on their body. Then there is pili, sex pili, which is help in conjugation. That is movement of one genetic particle from one bacteria to another bacteria through sex pili. Okay, these are the function. Then how to demonstrate fimbriae and pili? Demonstration by our normal, one method is our normal electron microscopy. Then the second one is by surface pellicle. So what is mean by surface pellicle? Surface pellicle, when we culture the fimbriae containing microorganisms in a liquid culture media, then there will be this culture broth here, and in surface there will be a sluggish appearance of a pellicle. This is called a surface pellicle. If we can demonstrate surface pellicle, that means that culture media containing microorganism has fimbriae or pili. Okay, so this is all about cell wall appendages. Then nextly we are going to talk about is spores, and the fourth one is spores. So what are spores? Spores are the dormant form of bacteria. Okay, when unfavorable condition is there, when there are lack of nutrients, the normal vegetative state of the bacteria goes into a dormant form, that is called spores, and the process is called sporulation. Okay, this is a vegetative state, and this is the spore state. So this formation, that is from vegetative to spore state, is called sporulation. Okay, this method is known as sporulation. This undergo in unfavorable condition, and when favorable condition returns, then this dormant spore state will form backwards into vegetative state. This is called germination. Okay, so spores form under unfavorable condition, highly resistant. Okay, now let us discuss about the structure of spores. When we talk about structure, there is a core, central core. Okay, there is a core, cortex and coat. So there is a core, central core, and covering the central core there is a inner membrane. The second layer is the inner membrane. First one is the core, then there is inner membrane. This one is the inner membrane, and there is above inner membrane there is germ cell wall. This is the third. Okay, then there is cortex. This one the fourth. Okay, then there is outer membrane. Above cortex there is outer membrane, then above cortex there is coat, then above all the last layer is exosporium. exosporium. Okay, so it is easy. Core, cortex, coat. Okay, in between core and cortex there is inner membrane and germ cell wall. In between cortex and coat there is outer membrane, and above all there is exosporium. Then the spores can be in various shape. Okay, if we take a bacillus, the spores can be here, oval, round shape, various shapes can be there, and also their position can be different. It can be centric or terminal or subterminal. Okay, and they can also be bulging or non-bulging. This is a non-bulging spore and this is a bulging spore. If the spores comes out like here, this is a bulging spore and a non-bulging spore. For example, of non-bulging spore, this is bacillus, non-bulging. And if for example of a bulging spore, our Clostridium. Clostridium is example for bulging spore and non-bulging spore, and the spores can be either oval or central, or and also can be terminal or central or subterminal. Okay, so these are talking about spores. Then next let us discuss how we demonstrate spores. So firstly, spores are done firstly by our normal gram stain. We already saw that in the gram staining, spores are non-stainable, will not take stain, not take stain. So spores will not take stain, the rest of the organism will take stain. Okay, then there is another method is acid fast. So spores are slightly acid fast positive, so there will be reddish color, reddish or pink color. That's slightly acid fast, so a slight color will be only there. Then another method is special staining, that is called Fulton stain. Fulton stain and Moeller stain. These are the special staining. Okay, special staining technique for endospore. So this is how we demonstrate spores by staining. 1 gram staining, they are will not take gram stain, both stain. Okay, so they are acid fast positive, slight acid fast positive, and there are special stains like Fulton stain and Moeller stain. Okay, so next let's talk about the application. Application of spores. Spores are used as a material or a bioweapon. So in biowar, used in biowar, then there are used as control in sterilization technique. Okay, they are used as a control in sterilization technique. This for example, spores of geo, okay, sorry, Geobacillus dermo, sorry, Geobacillus. The spores of Geobacillus chiarodermophilus is used in our autoclave method and hot air oven. Sorry, not hot air oven, plasma sterilizer and plasma sterilizer. Okay. And the next spores we use as a control is Bacillus atrophius. Is used in hot air oven and ethylene oxide sterilizer. So you see Geobacillus stearodermophilus is used. The spores of Geobacillus stearodermophilus is used in autoclave and plasma sterilizer, and Bacillus atrophius is used in hot air oven and ethylene oxide sterilizer. So these are the two applications of spores. For example, in BioVar we uses Bacillus anthracis. Okay, spores of Bacillus anthracis. Okay, maybe you might heard about the Bacillus bioterrorism, anthrax bioterrorism. Okay, so in BioVar we use it as a bioweapon and as a control in sterilization technique. These are all about spores.